Okay. I'm glad to introduce Dr. Martemyanov. He is the chair of Neuroscience Department and professor at the UF Scripps Institute. His research focuses on the fundamental mechanisms of neuromodular G protein signaling in the nervous system, how it releases to neurological and psychiatric conditions, as well as developing new therapeutics.
Thank you so much. It's really my pleasure to be here. Hopefully everybody is now well caffeinated, which, you know, is one of the good treatments. And I guess pastries contain a lot of zinc as well, right? So I think we should be all set. Thank you very much for having me here. It's really, really wonderful to see this event play out another year. I'm really thankful for the community of patients as well for the support. There's been quite a bit of evolution of our thinking about GNAO1 disorder. One of the benefits of going almost last is that I'm not going to probably introduce many new concepts. I'm going to go through a lot of things that you've heard, but I'm going to give my spin take and sort of conceptually how we think about it. So there's been quite a bit of evolution from the original description of this, where we kind of thought that they're like two maybe distinct disorders, depends on which mutations you have. Is it more like a movement disorder versus epilepsy? But then with the advent of more and more studies as we get to quite a number of the mutations, which is almost like 58 mutations, that cover quite a significant number of patients, almost 200 of them. There's this wonderful paper that many of my colleagues here in the audience were an author on, has done a fantastic work summarizing what the current state of knowing the mutations there. And I'm picking up the basically the stats from this review, which essentially makes a very strong point that GNAO1 is in its essence is a movement disorder. If you count the number of patients, the percentage of patients that suffer from a movement disorder, that's actually 90% of patients. And another very frequent comorbid condition is, of course, epilepsy. All right, so we've studied G proteins and GPCRs for a very long time. So this is what my lab does. And we've studied all sorts of different G-alpha subunits for more than 20 years. So this is really the field that I'm very comfortable with, but I'm just going to give you a bit of a flavor of what those systems do. There's essentially the biggest protein family in our genome's period. This is by far the most prevalent cell surface receptors that are there to transduce any possible signals. This is basis for the neurotransmission and neuromodulation. And consequently, they're targeted by the largest fraction of the drugs currently on the market. Well, that makes things a little bit problematic when you're trying to understand what exactly goes wrong with the mutations in the GO. But one thing to know is that GO is just one of the alpha subunits of the G proteins. There are 15 others. So it's a bit of a complicated way of how those GPCRs transmit the signal. But invariably, they need two components. It's a beta-gamma subunits that you've heard of, and it's the G-alpha subunit that is utilized to interact with the GPCR that gets activated, and then both of them are sent apart to transduce the signal. So it's beta-gamma transducing the signal and also the G-alpha. And in this case, we care about the G-alpha O. So here's just a very much simplified scheme of how you transmit the signals of the GPCRs in general through the G proteins. You can have either stimulatory signals that are sent through the G-alpha S OFF types of the G proteins that typically stimulate cyclic EMP production. They have like other targets as well, or inhibitory that typically signal through the GI types of the G proteins that also release the beta-gamma subunits and negatively control cyclic EMP production by the cyclases. There are also regulatory proteins, RGSs that I've heard from, that negatively regulate the GI and a GO branch of the signaling. So what's interesting about the scheme is that in the past 5 years, nearly every component in this scheme is become a movement disorder. So we've done original work on the GNL, which is encoding G-alpha OFF. EDC5 is another disorder. The mutations in GNB5, which is component of RGS, gives another movement disorder. And GNB1, that you've also heard, is a movement disorder. And very recently, mutations in the G-alpha I also became a movement disorder. So the question is, what is the G-alpha O does in this scheme to regulate the cyclic EMP production and balance the signaling to get to the movement disorder as well? So we're in Rome, so I thought that we're going to talk about 3 pillars that I think are important for understanding GNAO1, which in my opinion is GPCR system involved, the brain region that's involved in a disease, and a mutation mechanisms. And I try to touch upon every aspect of this. So let's start with the GPCR system involved. This is part of the unpublished work that we've done, and I'll introduce the system that we use to evaluate the GPCR signals that we've developed a few years ago. We essentially use the power of bioluminescent resonance transfer to study the activation of the G proteins. And in which case, we're not modifying the G-alpha subunit. We essentially tag the beta-gamma subunit with the fluorescent protein Venus to study its release. And when it gets released, it binds to the reporter for the beta-gamma called GRK, and that's tagged with luciferase. So we can basically monitor the breath signal development and deactivation as the G proteins get activated or deactivated. So we've been busy profiling the activity of many different GPCRs. To this date, we profiled more than 100 GPCR activities on different G-alpha subunits. Remember, there are many different G-alpha subunits. And what we found, and that's basically, you can't discern much, but the first column is actually the GO, the ability of the GPCRs to activate the GO. As you can appreciate from this brief graph, essentially 80% of GPCRs can activate the GO. So you can appreciate if the GO cannot function very well, you're not affecting just one GPCR, you're affecting the vast majority of the GPCRs that now would be incapable of transmitting the signal. So let's get briefly through the mutation mechanisms that have been figured out. I already introduced the system. It's actually a very powerful system that we can take in a relatively high throughput to understand the G protein dynamics. It's based on breath. We can study not only the activation of the G proteins, but the dynamics of the alpha beta gamma association, dissociation, the trimer formation. We can actually look at the expression profile of the effects of mutations on expression, as well as we can study the propagation of signal downstream. We can look at the second messenger, such as cyclic EMP, directly. So we've historically evaluated a few mutations. That is, what you see on the left is part of what's published. And more recently, in a collaboration with Dario Escobar, we've analyzed also the Spanish cohort of the mutations. So if you plug in those mutants, which is like a couple dozen that we've already introduced in the system, you'd appreciate that every single one of them gives us a loss of function phenotype. So meaning that the mutations in this GO are not tolerated in terms of being able to transmit the information from the GPCRs. We've also probed the dominant negative activity, and there's a bit of a semantic of what you call dominant negative versus neomorph. I think we may be talking about the same thing, which is basically a toxic activity of the G-alpha when you introduce a mutation that makes it not only incapable of transmitting the signal, but interfering with its ability to activate normal G-alpha subunits in such a way that you cannot overcome it by getting more of a normal G-alpha subunit. This is exactly what we do. It's the same type of the assay. You just, on top of the wild-type G-alpha, you introduce a mutant copy, and for many of the mutations, actually for some of the mutations, when you do so, you see the inhibition of a signal transfer through the normal G-alpha subunit. That, in our opinion, is what we call the dominant negative effect, and Vladimir calls it neuromorphic mutations. So there could be multiple mechanisms for how this neuromorphic or dominant negative activity is manifested. We can look at the trimer formation, which is ability of the alpha subunit to bind to the beta gamma subunit, which is essential for the G protein activation and ability to transmit the signal. And in case of several of those mutants, we see the clear deficit, or sometimes actually the gain in beta gamma binding ability. You can also look at the ability of the heterotrimer to associate with the GPCR. And again, for some of those mutations, we see a clear evidence that we are dealing with a gain of function. Some of those mutations pre-couple the complex with the beta gamma and the alpha with the GPCR to a much greater extent than the wild type can. So you can imagine that this activity can actually clog the GPCR and make it unable to activate the normal types of the G-alpha subunits. All right, so Darius already introduced this a little bit, so I'm just going to go quickly through our functional side of the story. You can look at this dominant negative effect and quantify it, and also quantify the loss of function effect. And what you would see when you run this exercise, you would see that the mutants are actually stratified along two axes. They have a spectrum of a dominant negative activity, and they have a spectrum of the loss of function activity. So it's actually two-dimensional way of looking at how the mutations in the G-alpha-O would be perturbing. You can also come up with a severity scoring for how bad the signaling disruptions are by a given mutation and stratify those mutants in this microarray-like format. And for each of the case, you can quantify actually the functional measure of how disruptive a particular mutation is in ability of a GPCR to carry the signal. And if you do this, you can pair it very nicely with a clinical severity score that Dario Escobar has developed and run a correlation, and that's actually pretty decent correlation between the experimental score that we observe in our system of assays and a clinical findings. But we have different measures that we evaluate in our functional assays. So we can run those correlations between different measures, and one of the best correlations that we get of the clinical severity is actually with the formation of the trimer assembly. That's the interaction between the alpha subunit and beta gamma. So there's something fundamentally off when this mutation is introduced and it binds in with the beta gamma subunits, which is actually pretty interesting because I don't know how many of you have seen this paper. This is a fairly recent paper that doesn't really deal with the mutations in GNAO1, but in effect it does. So this is a paper from Henrik Dolman's lab who's looking at the mechanisms that alpha subunits are utilizing to keep and release the beta gamma subunits. I was actually asked to write a news and views on this paper, just go through the findings that were found in this work. And it's essentially illustrated here. There are three residues that form the allosteric channel that control the nucleotide state. And those three residues are outlined here. It's the conformational rearrangements that occur upon the nucleotide binding that are mediated by these three residues that are essential for the trimers to go through the rearrangement that leads to the beta gamma release. And one of those residues is glutamic acid 246, and other one is arginine 209, and yet another one is glycine 203. Those are the most prominent GNAO1 mutations. So I would submit that it's fundamentally the ability of the heterotrimers to undergo through this conformational change that releases the beta gamma subunit that is key to the pathology of this disorder. All right, so Simona has been very kind introducing the model for the C. elegans. I'm not going to belabor this that much because we need to test a very critical assumption that some of those mutations just loss of function versus is just a loss of function and encumbered by the dominant negative activity. This is what we're going to be testing using this very powerful model of C. elegans. And it's been a very classical model to study GNAO1 for many, many years. So if you have a gain of function in GO, these worms are hypoactive. When it's a loss of function, they're hyperactive. You can easily quantify their behavior, and you introduce a model mutation that leads to the gain of function because they simply can't hydrolyze the GDP, and which we've done in this experiment, and also loss of function, which is essentially a knockout of a GO. Very easy to tell whether that's a gain of function versus loss of function. Then you use a CRISPR method to basically mutate this to the G203R, R209, and we've done the 42R as well, and you can clearly see that all of these mutations, when introduced in both alleles, clearly give you a loss of function phenotype. Well, Simona has also introduced the LG-carb assay. You can actually assess now the sensitivity of these worms to the paralyzing effect of the LG-carb. And if you do this just by CRISPRing two alleles, you can see that all of us mutants are simple loss of function, but then you can modify this assay and actually introduce mutation only in one allele and keep another allele wild type, which is what you see in this gray bars. When you do this with a wild type, just reduce one allele, worms don't really care that much. They still have the same sensitivity to LG-carb. But when you introduce the point mutations in the second allele, while you keep in one of the wild type, you see the same loss in the sensitivity to hypersensitivity to LG-carb, which basically means that all of us mutations that we evaluated are dominant negative in the C. elegans. All right, so I'm going to deal with the brain region aspects of this. What would be the good brain region to look at? And the place where we like looking is a striatum, which essentially is the key brain structure to control movement and involved in a variety of movement disorders from dystonia, Parkinson's, to Rett syndrome. Dopamine is one of the key modulators in the region. It just makes perfect sense to look in the striatal neurons, not to say that this is the only place that's relevant for the pathology. It's just certainly involved in the movement disorders. So we've generated a loss of function model for this that's published by now. It's just elimination of a GO. And this is a floxed allele of a G-alpha-O gene, GNAO1. We cross it with a Cre driver line, RGS9 Cre, which is selective for the striatal neurons. And then we have our panel for the evaluation of the mouse behavior, which we call like a little dystonia movement sort of clinic for the mice. And we can look at the clasping scores, the limb coordination, when you force the mice to walk backwards, the dynamic balance, which is basically a wire on a ledge type of the score, and then a motor learning on rotor-rod. And when we evaluate this mice, we essentially come with a uniform conclusion that they are deficient in the motor domain all across this evaluation criteria. We can also probe the dominant negative effects by introducing the viruses that encode the pathogenic mutations through the AAV in the wild-type animals, and animals that have the Cre recombinase expressed in either direct medium spiny neurons or indirect medium spiny neurons. And when we do so, we also notice the very similar movement coordination problems across the domains that we score. So it essentially shows you that just having a dominant negative effect through this pathogenic variants also gives you the phenotype. All right, very recently, we've been involved in developing the model for the dominant negative genetic model. And this one is taking advantage of G203R knock-in. As many of you know, and Vladimir showed this beautifully, these mice don't actually survive. So we decided to take a different strategy for developing this mice and making actually a conditional knock-in. So it's a bit of a complicated design. We essentially introduce the point mutation G203R in exon 6, and then precede this with a cassette that contains the cDNA for the remainder of the protein that's going to accept the splicing and going to create the wild-type allele. And we flux it, and also in inverted direction, we introduce this selection cassette with a marker, essentially, which expresses TD tomato in every neuron where this modification happened. And then when you introduce the Cre, and in this case, we cross this animal model for the validation studies with the same RGS9 Cre, it's going to cut at the flux sites, restore the normal gene architecture, except that now we'll have the G203R mutation. So that's going to be a conditional knock-in. So with very preliminary, this is very hot off the press. This is essentially 2-week-old data. We've started looking at the brains of these mice, comparing the Cre positive and the Cre negative animals. DAPI basically stains all of the cells. TD tomato is a reporter just to basically show that we can, the genetic recombination is successful. We created this allele, and you see red everywhere. That basically illustrates that, you know, this cassette made it to all of the cells. You see very prominently blood vessels. So this wiggles here is essentially blood vessels that are now fluorescent. So, but we will magnify this region, and when you magnify the region and start comparing the Cre negative to the Cre positive, blood vessels stay red, but all of us neuronal cell bodies that you see as dots, we eliminate the TD tomato, essentially meaning that we can effectively excise the cassette and introduce the G203R knock-in. This might survive. So we started evaluating the motor coordination, and we've run the open field. We'd expect this mice to be hyperactive, and we do, in fact, detect quite a bit of a hyperactivity in the open field test. We've also run the rotor-rod, and what the rotor-rod shows us is essentially this mice cannot learn the task completely. It's basically a bicycle, you know, learn to ride the bicycle type of the assay. You see that in the presence of the Cre recombinase with a point mutation just in the striatal neurons, this mice can learn the task while wild types can't as a Cre negative littermates. So we're certainly interested in furthering this studies, and we have like quite a number of different lines to cross this line with. We want to cross it with a constitutive brain wide. We'll see if this might survive. We crossed it with a chem kinase excitatory for brain neurons. We'll see if these guys survive. And then we can cross it with inducible cell lines that you need to administer with tamoxifen that's going to allow us to define the developmental window for manipulation. So go back to this cascade, just a couple slides I run through before I end. So we also want to see what impact the mutants make in the cyclic AMP generation, and we've developed a mouse model for this as well, which is a fluxed animal to introduce the thread-based reporter that allows us to study the cyclic AMP production in real time. This is published. I'm going to go very quickly through this. We've looked at the aspects of cyclic AMP regulation when you stimulate with dopamine in both the D1-MSNs and I-MSNs. And what we see in the absence of a GO, this is loss of function model, you see a prominent leftward shift. There's more cyclic AMP production. In response to the GPCR stimulation, you have now more effective GPCR signaling through the stimulatory action of a GPCRs. And indirect MSNs, you actually see blunted inhibition. So that basically creates the perfect storm of more effective GPCR signaling when you lose the GO function. So what G-alpha-O does, in my opinion, is essentially controls the sensitivity of a GPCR signals to the cyclase and the cyclic AMP production. It stimulates the GS signaling in the absence of G-alpha-O. It inhibits the inhibitory. So the loss of GO function is going to affect fundamentally how much of the cyclic AMP is produced. And that in turn affects the synaptic plasticity, excitability, and generation of the neuronal responses. With this, sorry for going over time. I just need to thank the people who've been involved in this work. These names are highlighted in red, and thank you very much for your attention as well.
Thank you so much. We didn't want to pressurize you.
I keep losing this.
It's okay. Thank you. This is very interesting, and a new mouse model, difficult to build up, as you said, but eventually it's going to provide a very good model to study this further. Okay, are there any questions? Vladimir, please.
It's marvelous, the model you have created. Can you please remind what was the, so the Cre you used and you showed, is it early, is it regional, is it pan-neuronal? I didn't catch it.
So RGS9 Cre that we use is striatal-specific Cre. It's active mostly in the striatal neurons, some hypothalamic neurons, but it's like considered typically in the field striatal-specific Cre. It kicks in at postnatal day 14. Like as many striatal genes, the way they are regulated developmentally is they tend to kick in pretty late in neuronal development. So we certainly need to cross it with like earlier and define the window and all of that.
Excellent, thanks.
Other questions? Can I ask you? You mentioned about the specific tissue, which of course is very important, you know. So do you expect, tissue-specific alterations also at the transcriptional level? And would this model be an interesting model to do, for instance, a single-cell study in the tissue taken from the striatal area that you mentioned to see which type of neurons are indeed the most involved?
Yeah, so certainly there are pluses and minuses of conditional design, right? But the pluses of conditional design is exactly what you mentioned. You can very specifically, in a neuron population fashion, turn it on and see what impact that particular population makes. It's going to be a little bit further away from modeling of a clinical part of the disease, because now you're in a circuit-specific fashion, but it's going to teach you a lot about the fundamental biology and the contribution of circuits to the behaviors and what disruptions are there. But given the involvement of a GO in a variety of a GPCR's brain regions, I would expect that it's likely going to be detrimental in many brain
Yeah.
regions, not just the striatum.
Absolutely, yes. Any other questions? No? Okay, thank you very
Great, thank
much
you.
for your nice talk.